Hey, Mr. P here. This video is all about chromosomes. In our last video, we talked about genes, we talked about gene mutations, we talked about the alleles that make up those particular genes, and we talked about locations a little bit of genes on specific chromosomes. But in this video, we're going to dive into a more in-depth discussion of chromosomes. We're going to look at the differences among prokaryotes and eukaryotes in terms of their chromosomes. We're going to look a little bit more about the anatomy of chromosomes, and then a little bit about chromosome abnormalities and what happens not only if a gene is mutated, but also what happens if that mutation or what happens if that alteration is at the chromosome level. Okay, what happens if you have too many chromosomes? What happens if you have too few chromosomes? All of those things are going to be looked at. Stay tuned. Let's start off with a kind of in-depth look at, or a general overview of, the prokaryote and eukaryote chromosome differences. You'll notice that throughout IB biology there is much to talk about in terms of the differences among prokaryotes and eukaryotes, but in this particular topic we're going to look at just the differences and similarities of their chromosome. In a prokaryotic cell, which remember is a cell like bacteria, those cells don't have a nucleus. They don't therefore have a nuclear envelope. They don't therefore have a kind of specific place that encapsulates their DNA or their genetic material. They keep that chromosome in a nucleoid region, which I know we've talked about in previous videos, but the big difference between prokaryote and eukaryote chromosomes is the fact that prokaryotic chromosomes is singular, meaning prokaryotes have a single chromosome, and that single chromosome is circular. Okay, so it is circular, meaning it starts and stops, if you will, at the same spot. Okay, a difference among eukaryotic chromosomes is, one, the chromosomes are actually multiple. Okay, so there are multiple chromosomes associated with a eukaryotic chromosome. The chromosomes of a eukaryotic cell are kept or contained within the nucleus, which is the organelle that is double membraned. We call that a nuclear envelope. It's porous. That allows for the mRNA that is made or transcribed in the nucleus to get out. It also allows for ribosomes to get out. All of that has been talked about, but just for the sake of review, is thrown in. So right off the bat, eukaryotic, prokaryotic chromosomes, prokaryotes is a single circular chromosome kept within a nucleoid region. Eukaryotic chromosomes, there are multiple of them. They are kept within the nucleus, meaning they're more protected, and the eukaryotic chromosomes are linear. Okay, linear, meaning they are not circular. There is a definite start and stop. Okay, and eukaryotic chromosomes are typically going to be associated with that typical X shape that you've become kind of accustomed to in terms of chromosome shape. Prokaryotic cells don't make that structure because it is very difficult, impossible even, to take a circular ring of DNA and structure it in a way that allows it to be linear, kind of a linear X shaped like this structure. So this X-shaped structure is very much eukaryotic, and when you look at eukaryotic chromosomes, specifically eukaryotic nuclei, you'll notice most of the time that there are multiple chromosomes in a eukaryotic application. Most of the time in diploid organisms, these chromosomes will be paired in homologous pairs. We'll get to that in a little bit. Prokaryotic cells obviously are not going to have homologous pairs because of the fact that they have a single circular chromosome. Prokaryotic cells also do not have DNA that is associated with histones. You'll notice that on this eukaryotic side there are these histone proteins, which we've talked about in previous videos as well. These histone proteins are going to orient themselves into an octamer of histone proteins called a nucleosome, and each of these octamers are going to be encapsulated or surrounded or wrapped with DNA twice. This is a nucleosome, and those are going to be instrumental in organizing this really big, long, linear molecule of DNA so that it not only fits into the eukaryotic nucleus, but it actually allows for the gene expression to be kind of regulated in a eukaryotic application as well. So, prokaryotic cells, circular ring of DNA, singular, not associated with histone proteins, so we say that the prokaryotic DNA is naked. Kind of a weird term to use, but it is definitely something that is used. Naked just means that it is not associated with histone proteins. Obviously kept within a nucleoid region, no nucleus at all. Eukaryotic cells, just to kind of wrap up, there are multiple linear chromosomes. Those chromosomes are kept within a nucleus, and those chromosomes are typically paired in homologous pairs, at least in diploid organisms. And the DNA is not naked. It is associated with histone proteins, and those histones are wrapped into or grouped into octamers called nucleosomes. So, let's wrap up what we just talked about, at least in kind of word form. We've got prokaryotic DNA specifically. The DNA is kept within a circular chromosome that is singular. Okay, a single circular chromosome. It is naked, not associated with histones. Okay, and one thing we didn't talk about, but you can definitely see it here, prokaryotes typically have plasmids. You can see it right here. That is not the nucleoid region. Okay, you'll notice that most prokaryotes are going to have their main molecule of DNA kept within this nucleoid region. It contains the genophore or the genotype, the genome of the particular bacteria cell. But there is a separate, smaller ring of DNA called a plasmid, and the plasmid is going to contain several survival characteristics, including antibiotic resistance in a lot of cases. Okay, these plasmids have a lot of biotechnical applications. We'll get to that in future topics, but at least at this point you need to know what the plasmid is, where it's located. It is located within the cytoplasm, definitely outside of the nucleoid region. It is not associated with the actual genome of the bacteria itself. It is, however, still naked DNA, not associated with histones, and it is still singular and circular. It's just much smaller and kind of a secondary bit of information that contains survival characteristics like antibiotic resistance, and that particular plasmid or that particular ring of DNA can actually be passed between prokaryotes. So from prokaryotic cell 1 to prokaryotic cell 2, it is passed through a conjugation pilus in a process called bacterial conjugation, which is kind of analogous to sexual reproduction in a prokaryotic application. We know at this point, or hopefully you know at this point, that bacteria cells can't sexually reproduce like eukaryotic organisms can. However, they can influence each other's kind of offspring, or they can influence each other's replicates by passing on particular information like antibiotic resistance from bacteria cell to bacteria cell through a process of conjugation. The bacteria plasmid can be incorporated into the nucleoid chromosome upon conjugation. So if a cell receives a plasmid from another cell, it can then incorporate that bit of information into the nucleoid chromosome. And this recently, like I said, became useful in biotech applications as a way of producing human proteins without actually sourcing those proteins from kind of unethically sourced human applications. One of the ways that these biotech applications work is that we take a plasmid and we use the circular ring of DNA kind of as the vector by which we will not only target the gene, but will also ensure that the gene is expressed within the prokaryote that receives the plasmid. And the nice thing about the DNA being kind of that universality or the universal nature of DNA is that because DNA is created from A, T, C's, and G's. All living things that are DNA based have the machinery and the cellular machinery to not only read the DNA, but to also express and produce the proteins through the production of mRNA that the particular organism needs. And so what we have done in biotechnical applications is we have created a plasmid that includes a target gene, in this case human insulin, so that when we as scientists or biotechnologists insert this plasmid into, most of the time, E. coli cells, which definitely is a prokaryote, the prokaryotes, or the E. coli in this case, will produce or begin to produce human insulin because of the fact that they now have the target gene for the production of insulin in their nucleoid genome. And when they start producing human insulin, we then take that cell that's producing human insulin and we can isolate and purify the human insulin that the E. coli cells are making, and then use it in medical interventions for individuals that are insulin deficient or who can't otherwise produce insulin on their own, diabetics and so forth. So cool use of biotechnology. It's a cool use of our understanding of DNA. It's a cool use of our understanding of plasmids. Definitely a win-win for everybody. Let's dive in a little more about the eukaryotic chromosome. We've talked about all of this in that previous slide. However, I like to kind of show it in multiple ways. And so, again, that eukaryotic chromosome is typically paired in diploid organisms. Okay, you'll see here that it is shown as a pair. You've got the maternal homolog, you have the paternal homolog. Paternal obviously would have come from the inheritance from your father, or would have been inherited from your father. Maternal would have been inherited from your mother. The chromosomes in a eukaryotic application are going to be X-shaped. The X-shaped chromosome is going to be condensed DNA molecule that is wrapped around those histone proteins that are grouped in the octamer called the nucleosomes. They're going to come together in this X-shaped to produce two identical sister chromatids. This would be chromatid 1, this would be chromatid number 2. The X-shaped chromosome is going to be held together by a central protein complex called a centromere. Two additional components related to the centromere is going to be these little protein complexes on the centromere called kinetochores, which are going to ultimately allow for the attachment of the spindle fibers. But when we hold two sister chromatids together by a common centromere, it produces these upper two arms and these lower two arms. We call the upper arms P arms and the lower arms Q arms. The Q arms are always the long arm structure and the P arm is always the short arm structure. Okay, so in this particular chromosome and in most chromosomes, you're going to have a short arm structure and a long arm structure. Here you can definitely see that is the centromere. It's the point in which it pinches in. These on top would definitely be the short arm structure, so we would call that the P arm. These are the long arm structure of this particular chromosome, so this would definitely be the Q arm structure. And you will also notice that on these paternal and maternal homologs, meaning one member of the homologous pair versus the other member of the homologous pair, they are not identical to each other in terms of the actual alleles. Now, in this particular gene locus, there are two identical alleles. This individual would have received a small r from the father and a small r from the mother. So that is a homozygous recessive gene locus, okay, or homozygous recessive alleles at that particular gene locus. You can see that these terms down here, okay, if it's a little, it's a homozygous recessive application. If it's a big, it's a homozygous dominant application. If it is a big, it's a heterozygous application. So these are three different genes at three different gene loci or gene locations. But you'll notice that as you go down this particular Q arm, there is not only an R gene location, there's a P gene location, A, B, and C. Each one of those is a different set of alleles for a different gene at a different gene location. And you will see that several of them are either, homozygous dominant, like the P's and the A's. 2 of them are homozygous recessive, like the R's and the B's, and 1 is homozygous, like the C's. Homologous chromosomes carry the same sequence of genes, but not necessarily the same alleles of those genes. It is important for you to know that the homologs, meaning the homologous pair, are not identical, even though they carry the same sequence of genes. Basically, we can say that both of these homologs are identical in the terms of which genes they possess, or which genes they carry. However, you'll notice that the specific alleles of the genes they carry are different, and so therefore the actual sequence of nucleotides differs among the homologs, and the information that is therefore coded for is different in both homologs. Eukaryotic chromosomes vary by length, position of the centromere, and genes found at specific locations within that particular chromosome. And so you'll notice that this is the entire genome of a human. This isn't a specific genome of a human, because you'll notice that down here in this 23rd pair location, it gives you an either or. Okay, this on the left would indicate a male, and this would indicate female. The way that you know that is females are XX on their 23rd pair, and males are XY on their 23rd pair. We'll get into more of that in a minute. But we have, as humans, 23 pairs of chromosomes, which means we have 46 total chromosomes. If you had 23 pairs of shoes, you would have 46 total shoes. Okay? You'll notice that each one of these chromosomes, which are kind of denoted in this particular diagram as different sizes, and that's by design. Okay? 1 is the biggest chromosome, 2 is the second biggest, 3 is the second biggest, and so forth. You'll notice that the centromere location, which is indicated kind of by this dark section, changes depending on which chromosome we're looking at. Obviously, 1 is kind of more centrally located. 2 gets a little bit more towards the top. Some of them, like 13 and 14 and 15, are very close to the end in terms of their chromosome or their centromere location. You can see that the length of the chromosome differs as well. Okay, some chromosomes like 21, 22, and even the Y chromosome are very short. Some chromosomes like 1, 2, and 3 are very long, and so the length is dependent on the specific number of nucleotides in the DNA molecule. The more nucleotides a chromosome contains, the longer the molecule will be. Okay, and when I say the number of nucleotides, I'm talking about the number of A, T, T, C, G, okay, or A, T, C's, and G's. It's literally the number of those letters that indicates the length of the chromosome. Okay, the more letters you have in the DNA sequence, the longer the chromosome has to be in order to maintain all of the information that that particular DNA sequence has. Position of centromere, there are 4 types of centromere locations: metacentric, submetacentric, acrocentric, and telocentric. Okay, and it is really important for you to know that metacentric basically means middle. So a chromosome that has a centromere position in the middle, or meta, meaning middle, that is a metacentric chromosome. Submetacentric chromosome is basically almost in the middle, but just slightly off to one side, like this. Okay, it's a submetacentric. Acrocentric is almost to the end. You can see that here. Okay, it's almost on the end, but not quite. Telocentric would be completely on the end. Okay, like there. So, metacentric in the middle, submetacentric slightly off center, acrocentric almost to the end, telocentric actually on the end. Genes found at a specific location. You will also know that the banding or sequence, the banding sequence of all these chromosomes are different. That banding kind of just indicates the types of genes and the number of genes located on each of these chromosomes. Not all genes are the same on each of the chromosomes, but they're all located in the same location among homologs. Okay? So, all of these chromosomes are different. They all are different based on length, position of centromere, and the genes on that specific location. This, while I have this kind of on the screen, is called a karyotype or a karyogram, and this type of image is used to map homologous chromosomes in descending order by length, which typically, like I said, goes numerically as well. They are visible in a eukaryotic nucleus at the beginning of the cell division, mitosis. Remember, the only time in a cell cycle where chromosomes are visible is in times of active cell division. Chromosomes are kept within the chromatin format throughout G1, S, and G2. They are only going to be supercoiled into or condensed into the X shape as soon as cell division or mitosis takes place. Okay, so different chromosomes equals different genes. We've kind of identified that. I talked about how different gene locations exist on different chromosomes. I have two examples here. I have chromosome 9 and chromosome 22. You'll notice that when you look at a karyogram, it goes basically chronological order, but it goes in descending order as well. So chromosome 9 is bigger than chromosome 22. We talked about the size of the chromosome is dependent on the number of base pairs, and so you can see that this definitely reflects that. 136 million base pairs specifically on chromosome 9, 49 million base pairs specifically on chromosome 22. But you'll notice that all of these fine prints are specific genes or specific traits or specific disorders that have been linked to these particular chromosomes in these particular locations. One of the big kind of aha moments or the big wins of the Human Genome Project is not only did we sequence the entire human genome, but we also learned a lot about the locations of genes, the size of chromosomes, the size of genes, the location of genes, stuff like that. So, eukaryotes possess multiple chromosomes. Multiple chromosomes are different sizes and centromere locations based on the number of base pairs present, but they each have specific gene locations with specific base sequences that code for different traits and disorders. Now we need to talk about haploid diploid or ploidy, if you will. Two terms you need to be familiar with, haploid and diploid. Haploid is used or uses the symbol N. Diploid uses the symbol 2N. Haploid refers to the number of unique chromosomes possessed by a species, and diploid refers to the number of duplicated chromosomes possessed by a species. Okay? The haploid number in humans is 23, which is why we, as a diploid organism, possess a diploid number of 46. Okay? How do we get our haploid number? We receive a copy of our mom's chromosomes and a copy of our dad's chromosomes through our inheritance and through sexual reproduction. Mom's gametes, the egg cells, have 23 chromosomes. The dad's sperm cell has 23 chromosomes, and when those come together to form homologous pairs, it produces 46, or it produces a cell called a zygote with 46 total chromosomes. Okay, haploid basically just refers to, like I said, one copy of each chromosome, and then diploid, once each of those chromosomes pair up into the homologous pairs that happens as a result of sexual reproduction, then we become our diploid number of 46. Okay? How does ploidy work? I'm not going to get into a really in-depth discussion of both spermatogenesis and oogenesis. That's coming later in the class, but you need to know that through the act of meiosis, which is coming at a later time as well, we can successfully take a diploid cell, which is 46, we know that because we're diploid organisms and our diploid number is 46, and we can, through 2 rounds of reduction division, take that 46 chromosomes and reduce them to 4 unique individual gametes that each have 23 chromosomes each. In spermatogenesis, like you can see here, we take a primary spermicide which has 46 chromosomes. It's a diploid cell, and we cut it down into 4 unique sperm cells, each with that magic 23 number. In oogenesis, again, it's the same process. It's basically meiosis. It's unique to females, though. They take 46 chromosomes in their diploid cell, which is a primary oocyte, and they cut it down to, in this case, 1 ovum. But again, it has 23 chromosomes. When you go through the process of sexual reproduction and specifically fertilization event, you will take 23 chromosomes from the sperm cell, you will add them to the 23 chromosomes of the egg cell, and it produces the first diploid cell, which is called a zygote, which is the first cell of the next generation, or the first cell of that particular offspring. Chromosome comparison. So one of the applications of IB in topic 3.2 is to look at different organisms and compare the number of chromosomes present in their genome or in their cells, to kind of look at a comparison of the organisms to draw any kind of unique ahas or takeaways from this particular lens. And so if we look at them, humans, like you and I, have 46 chromosomes. Asian rice, or Rhiza sativa, is going to have 24 chromosomes. They have roughly half the number of chromosomes we do. Dogs, or the domestic dog, Canis familiaris, is going to have 78 chromosomes, or almost double what we have. The chimpanzee has 48 chromosomes, and then if you look at the parasitic roundworm, which is equine roundworm, it's a specific roundworm in horses, is going to have only 2 chromosomes. Okay, so you might look at this and say, well, there is a variety of chromosomes. We've got organisms on the planet that have as little as maybe 1 or 2 chromosomes, as indicated by this parasite. We have maybe organisms on the planet that have as many as 78. Can we draw any kind of conclusions from this? The answer really is there isn't much correlation between organism size and number of chromosomes. There really isn't a correlation between the complexity of organism and the number of chromosomes. There isn't an indication of like ecological success and the number of chromosomes. Okay, it really is kind of random, and you'll notice when you look at even or expand your chromosome comparison, there are a variety of plants that have lower than 24, and there's a variety of plants that have higher than 24 chromosomes. There are a variety of mammals even that have more than 78, and there are a variety of mammals that have way fewer than the 46 we have. So it's random. It really doesn't matter. Okay, so sex determination, kind of coming to the end of this particular topic. We talked a little bit in our original karyogram about what determines gender, and what determines gender is the SRY gene. The SRY gene is a sex-determining region on the Y chromosome. It is linked to the Y chromosome, so it is only present in males. Okay, females are going to lack the Y chromosome entirely, and if you lack the Y chromosome, you're going to obviously lack the SRY gene, which is specifically located on the Y. The SRY, or the sex-determining region on the Y chromosome, is going to be linked to testosterone production, or an increased testosterone production, which is obviously going to increase the sexual characteristics and the maleness type traits, or the traits that are really related to males and being male. The presence and expression of the SRY gene on the Y chromosome leads to male development. I talked a little bit about that. The absence of the SRY expression allows the default gender or female state to develop. Okay, it is impossible to tell the gender of a newborn baby, or not newborn, but a fetus in fetal development through the first 20 weeks or so, because through the first 20 weeks, this SRY gene hasn't had time to be expressed yet. And so everybody, every fetus through the first 20 weeks is really kind of on that generic default female state. It's not until week 20 where the SRY expression really takes hold and starts to develop some of the external male characteristics. Early in development, the immature gonads of males and females are indistinguishable, and that is, like I said, through about 20 weeks. Okay, once that 20-week mark hits, then the expression of the development of external male gonads can happen, and that can be determined on a sonogram to actually definitively conclude the gender of the fetus. Inheritance of the fetus, or inheritance of the gender, is going to be determined in human inheritance by the male. We haven't gone through genetics yet, but you can see that females, because they are XX, will produce 100% of female eggs, or their gametes, that possess an X, meaning they can only pass on an X. Males will produce half of their sperm cells with an X and half of their sperm cells with a Y, meaning they will produce females half of the time and they will produce males half of the time. When you get an X from the mom and a Y from the dad, you obviously will develop a male. When you get an X from the dad and an X from the mom, you will produce a female. Next, we're going to look at a comparison of genome size among among several organisms related to Ibesyllabus. One, humans. Obviously, Homo sapiens is going to be 3.2 billion base pairs compared to a canopy plant, which is 150 billion base pairs. E. coli is 4.6 million base pairs. Fruit fly is 130 million base pairs. And then T2 phage, which are bacteriophage, is 164,000 base pairs. The main thing IB wants you to decipher from these particular organisms is that there isn't a particular or any kind of direct correlation between organismal complexity or organism size and genome size. One of the skills is to use a karyogram to determine gender. So I have two karyograms right here. One on the left is a female. When you look at a karyogram, obviously you can see the homologous chromosomes as they are arranged in not only their homolog but in descending order. This individual has an XX, so she is female. This individual has an XY, so he is male. When determining gender, you just need to look at the distribution of the sex chromosomes, or the 23rd pair. If it's an XX, it's a female. XY, it's a male. Another one of the skills is to use a karyogram to determine disorders, or determine a disorder. I have two karyograms again on this slide. You can see that the karyogram on the left is different than the one on the right. This would indicate, or can indicate, a trisomy on the 21st chromosome, which indicates Down syndrome. And the the karyogram on the right is Klinefelter's, which is an XXY. Okay, two things right off the bat that you notice. Klinefelter syndrome, it looks like a male and a female. There's two chromosomes for the X, or a pair of homologous chromosomes in the X chromosome and a Y. That would be a trisomy in the 23rd pair. And then you have a 21st trisomy, which would be Down syndrome. Last thing we've got to talk about is the audio radiography, which is looking at a particular chromosome but looking at it through a radiographic imagery. In this technique, they use radiation from a substance that is typically an isotope, and they capture it by photographic film or some kind of camera sensor. They then look at the radiography, or the radioactive material, and where it's located in order to determine size and location of the particular DNA molecule. It's used in genetics and often gives detailed pictures that indicate size and location. It's also part of the cairns technique. which is a specific technique used to not only identify the length of DNA or the size of DNA, but also where particular DNA markers are located. They use a radioactive derivative of hydrogen called H-thymidine, and it is a specifically tritiated thymidine, and they use it to or expose newly synthesized or newly replicated DNA molecules to a particular radioactive isotope of hydrogen in order to kind of ensure that that particular newly synthesized strand has that radioactivity that they can then sense with their cameras and develop it with a photographic film. So not much you need to know about it. You just need to know what the cairns technique is and what it allows researchers to do. It's all about identifying length of DNA molecule and specific location of particular markers of interest. Okay, that's it. Bring your questions to class. See you next time.